have Professor Shuefen Wang that will share with us information on research and practices in the HLB management in China, bringing an international view on this topic. Professor Shuefen Wang is the director of the Citrus Research Institute with the Southwest University in Chongqing. He has worked for many years in the study of greening, especially in the plant-bacteria interaction and also in the disease control technology. Currently, he is a member of the Executive Committee of the International Citriculture Society and the Secretary General of the National Alliance to the Integrated Control of Greening. In the last years, he's been coordinating projects with the National Program of Research and Development of High Relevance and key projects in the National Foundation of Natural Sciences in China, and he's published more than 50 articles in important journals, including Science. Professor?
So, good morning, everybody. Yeah, it's my pleasure to be here because thanks Fundecitrus and Giuliano's invitation. You know, me and Professor Ye get a chance to be here. Well, we talk with Fundecitrus scientists and get to take around, you know, some big processing company, the Citrus Circle. And this afternoon we will be Culturus, Grover, maybe there. Yeah. So, well, for this presentation, I would just like give you a very brief introduction about on our citrus HLB management in China and a couple research on our group, because I don't want to take much more time. Yeah, maybe 15 minutes to 20 minutes. I would like to leave more time to Professor Ye. Well, our institute is okay. Okay. So yeah, wait a moment. Is it okay? Okay. So yeah. Just a brief introduction of our institute. You know, it's actually a national level citrus research institute in China, and we do from gene plasma maintenance collection is done work. Okay, so so the mission of the institute is very similar with Fundecitrus, is to maintain the sustainability development of the citrus industry in China. But here in Brazil, I think Fundecitrus do the same work here. Oh, close. Okay, sure. Yeah. So as you know, the HLB, was first reported in China is in mainland China is in 1919. But in Taiwan is in 1913. There, and you can see is in the coast area in Chaozhou, in Guangdong province there. And this picture just shows very early, for HLB epidemiology evidence is, performed by Dr. Chen Qibao. It's pretty early. It's 1938 to 1941. It's 4 years in investigation for the HLB's how is the survey, and its epidemiology in Guangdong area, in Chaoshan area, as I mentioned, earlier, is first found in China. Well, before the HLB, people see HLB as citrus greening. But, in Giuliano, visited us several times, and Jose Bové also visited. We just, for the name HLB, Huang Longbing, just in memory of Professor Lin, who is, the first guy who experimentally proved that the virus, the disease, could be transmitted, graft transmissible disease. At that time, things it might be virus. But after that, Beauvais and his colleague proved that it's bacterial. Yeah. Also, I would like to, introduce Professor Zhao. Actually, his visit in Brazil is, I think, is in 2006. 2006, maybe, yeah. The first HLB international conference held in here. And he gave a talk here, and he did a lot of work on citrus HLB control in China. But also he contributed a lot to, the global, like Brazil and other countries. He's passed away this August, in 92. In 92, yeah. Well, in China, we have, 4 citrus superior belt in China, and oops. And so far, the psyllids, the HLB is, you can see found in the coast area, mostly in coast area, and in Chongqing, in my city, is in here, This region, yes, so far is HLB free, and Hubei, is HLB free. But unfortunately, psyllids was found in this area last year. And, the first found is surveys found the psyllids was in orange jasmine, in flower markets. There might be, this kind of orange jasmine is were imported from Guangdong and Guangxi, the coast area, and so it's very hard time for China, because might be a couple years later, is no HLB free area in China. Well, so far you can see from this slide, yeah, the HLB occurred counties is increasing. But so far it's still under control of HLB, but the situation is getting better. Well, we know, Professor Ling has, proved that 1955 or 1956, he found the HLB, the disease. At that time, is a virus. They think the virus is a graft transmissible. So, we usually use 3 basic measures. Also, I know it's when first, when HLB was first reported in Brazil. Juliano, the guys visit China, and Professor Zhao, I told you, he also introduced the measures here. So I think you guys also use these measures in Brazil. Also have some case is very good here is navel orange in Ganzhou. I think some colleagues in you visit there, and so far it's pretty good. It's the biggest navel orange producing area in China. So I think the first challenge is, the northward movement of psyllids. The second one is that, the 3 basic measures, remove tree, control psyllids, it's good, no problem. But right now, it's harder to performance by the farmers. You guys don't want to remove the trees. I think it should be almost the same here. And some case, you guys use the antibiotic. It's like United States since 2002. It's very regularly used in United States, but in China, some spots, in some counties, you guys also use the antibiotic. Yeah. So the picture from this side you can see is the antibiotic application is the experiment is performed in last century, the 1970s. It's useful. Yeah, it's useful, but the residue, antibiotic residue, and the biosafe issue. So in China, Chinese government don't encourage use, but not 100% stop this issue use in China. So that's still a problem there. Well, based on this challenge, we mean the psyllids and people don't want to remove the tree. So we do research funded by the national the government. They have some project to support us to do something, and we do some faster the diagnosis just with AI. You use if you use WeChat, we have some app there. It just scan the symptom, the leaf. It will tell you if it's infected or not. It's very quickly. It's widely used. And the guys also use screen wall. It's not covered like a green screen house like United States, in Florida, very big cover. It just uses the 4-meter screen wall, and the psyllids can't move usually, can move to inside. So it's also used in in Guangxi province and the Hunan province in some area. Yeah, it's quite useful. Also, yesterday, we talked. You guys use yellow trap. Yes, usually in China also use yellow trap. But my question is, who will check the trap day by day? So we collaborate with Huazhong Agricultural University in Wuhan. I guess now have an instrument, yes, for smart real-time the monitoring, and just integrated some instruments with yellow trap, with some lures, and you have camera, and you have AI technology to monitoring the psyllids. So it's very quickly you can respond. We can see it in your cell phone. And we also do a lot of interaction work because it's a bacterial, ACP, and the citrus plants. We do a lot of work. Some work with collaborate with Professor Ye. I just show a quick look for it. Well, we do a lot of because the psyllus is not like a typical bacterial because it's unculturable so far. It's like type 3 or type 4, the secretory system. But the psyllus has type 1 and second-dependent secretory system, and this effector, they might be 80. Yeah, the effector might have some very important roles in regulating plant immunity and other functions. So we do a lot of work. It's some effects can regulate autophagy and promote psyllus infection. I don't want to in detail to show more just. And some effectors could manipulate autophagy, some vacuum or lysosome. It's same, not only in citrus plants, but also in psyllids. The SAP is the same, almost same function there. And they also some effects could modulate ubiquitin system. And Professor Ye will show you SDE5, which have some function to regulate the plant's function. So we also do, citrus ACP's genomic. So far, the genomic is the highest example, best coverage and integrity. So it's used for some genomic salivary secretory protein system analysis. We do it, and it's released. Yeah, right now it's widely used. And finally, we do genome editing, because in citrus, my colleagues have used CRISPR-Cas9 based technologies to create citrus canker resistance. The gene plasma is the first one. It's published in 2017. So right now we use these technologies for creating HLB resistance gene plasma. We do have some strains have been proved for just intermediate biosafety testing. It's not released like Professor Francisco said. Right now it can be used in field, but it's just released in the greenhouse in Guangxi. Yeah, so it cannot be used in China for this. Well, I think we have a lot of challenges, but I think still have a lot of opportunities. We are doing where we are going to do the culture. It's uncountable so far, so it's difficult to find some cure, some medicals. So we are doing it. But today I'm not sure anything. We did make a very good progress on it. I hope next time I will show for it. Yeah, and a small peptide or small molecules, Dr. Yer will show. And yesterday we talked about we make a great collaboration with Fundecitrus, do some non-transgenic genetic engineering breeding. So I think it's a very good match well between our institute and Fundecitrus here. So we hope, But finally, I would like to say, before a successful cure for HLB, I think some basic measurement should be, prioritized, especially in very, low incidence area. If you just 3% incidence, if you use I think it's remove the tree on time. It's my recommended, yeah. So finally, I would like to thank my collaborator and Dr. Giuliano, your old friend. And thank you all. Thank you for listening. Thank you.